Okay, our next speaker is Tandy Warnow.
Okay, thank you. So I'm going to be talking about actually 3 topics that are relevant to this group. So after hearing people's talks, I thought I would squeeze as much in as I could, and apologies in advance, for I will be speaking quickly. Okay, so phylogenomics, which is basically the combination of phylogeny and genome scale data, that's the context here. And I just want to point out that you can see that in this, this phylogenetic quote unquote tree, it's not even a tree. There are these horizontal gene transfers. So when we're talking about phylogenetics, we're going beyond just looking for phylogenetic trees. We're also going to be interested in recovering where there are lateral gene transfers. So a phylogenomics pipeline is actually pretty complex. It starts with thinking about what data you're going to want to look at, figuring out orthology, potentially doing multiple sequence alignments on each locus, getting gene alignments and gene trees, either then concatenating the alignments to get the tree, or using the gene trees to get the species tree, or doing a network. And then looking at branch support, estimating dates, and trying to use this to look at biology. And the issue is that everything you're doing up here affects this. What happens with your final outcome depends upon what you did earlier. So there's a real consequence to accuracy based upon how you handle those estimation problems. So I've worked on several international consortia trying to get phylogenies for the large data sets. With the planned Transcriptome project, the problems were very large multi-copy gene families, which meant very large multiple sequence alignments. And we decided in the end not to even look at the multi-copy genes, which meant that we lost a lot of our data. And then we get a species tree from this, but then we see that the gene trees are very different from each other, and it's consistent with incomplete lineage sorting. Similar thing with the Avian project, except that there wasn't as much GDL, so we didn't lose really that much data. But we still had a lot of gene tree heterogeneity consistent with incomplete lineage sorting. So these are problems that affect many phylogenomics pipelines. And today I'm going to focus on these 2 questions. How do you do multiple sequence alignments? Okay, this is something that's relevant to pairwise alignment, but it's more complex. How do you get good gene trees? How do you use these alignments and trees to get species trees? And I'm going to be talking about how to do that by combining gene trees. So 3 topics. But I want to point out that everything I'm going to be talking about is really kind of a statistical estimation problem, because in all of these problems, you basically pose a mathematical model of evolution, whether that's only substitutions or has insertions and deletions, that depends. But you also have how genes are evolving inside a species tree. Different kinds of evolutionary processes. The estimation of the alignments and the trees is a statistical estimation problem. So large data sets are difficult for very interesting reasons. One is just size. You can have many loci, you can have many species. You can have many individuals in each of your species. You could have missing data, you get a heterogeneity. If you're allowing yourself to look at genome scale data, you don't get nice data anymore. It's not like every gene has lots of signal. There's going to be difficulties. So the larger your data set, the more complex, the more heterogeneous. And many of the analytical pipelines are using likelihood-based calculations, whether maximum likelihood or Bayesian. And this means they have numerics issues, they have runtime issues, they have memory issues, but they also depend upon the model. If the model is a problem, then maybe everything is a problem. So these are sort of the context. So today I'm going to try to talk about 3 different things. Getting species trees from gene trees, that was actually what I said I would talk about, but probably most of you here are not actually trying to get species trees. So you might be interested in some of the other questions more so. Next one is how to get very large maximum likelihood trees. And here, what I mean is many, many leaves in your tree. Maximum likelihood on really large data sets is really not adequately solved. So I'm going to talk about what we're doing to try to make it more feasible. And finally, I'm going to start with something on multiple sequence alignment where I have a conundrum. So I have really good results to tell you about how to get species trees. I have new work on how to get large maximum likelihood trees. And I have a problem about multiple sequence alignment. So I'm going to end with the problem because that's probably the most interesting thing. Okay, so species trees. So we have, here we have 4 species, but even these 4 species, human, chimp, gorilla, and orangutan, are not that easy. Getting the true tree on just these 4 species is complicated. And it's complicated for multiple reasons, even if the only thing you want is the unrooted tree. We're going to be talking about getting unrooted trees rather than rooted trees because evolution is basically time reversible. So statistical consistency, a very basic concept, says that in a statistical estimation problem, if you can prove that your method will converge to the correct answer with probability going to 1 as your data goes to infinity, it's a statistically consistent estimator. So we want to know if our species tree methods are statistically consistent. So what's going to infinity perhaps is the length of the genome. So length of the genome goes to infinity. Can we prove that error will go to 0? The problem is that there are all these causes for heterogeneity across the genome. Incomplete lineage sorting is a population level process. It happens all over. As I showed you, it happened with the plant data set, the avian data set. Every paper you read about phylogeny estimation is going to show you this. There's also gene duplication and loss. There's also horizontal gene transfer. And when you have any of these, you have species trees and gene trees being different. Here's an example just with human, chimp, gorilla, and orangutan. Human and chimp sibling here, that looks right. Human and gorilla sibling here, that looks wrong. But it's not wrong. It's correct for the gene. It's just not correct for the species tree. You get different gene trees than the species tree. So there's this model where the species tree generates genes. The gene trees can differ from the species tree. The gene trees evolve sequences, and you get sequences at the bottom. What do you do with the sequences at the bottom? If you're a standard biologist, you just do concatenation. You take all the alignments for all the genes, you put them into one matrix, and you run your favorite method, maximum likelihood, distance-based, whatever. It's concatenation using maximum likelihood. ML in phylogeny does not refer to machine learning. It refers to maximum likelihood. So that's hierarchical model. But if you do that standard concatenation approach, there are 2 theorems that have been proven that show that it can be inconsistent and positively misleading, converge to the wrong tree with probability going to 1. And that the 2 theorems depend upon whether it's partitioned or unpartitioned maximum likelihood. So it can give you the wrong answer with probability going to 1 theorem. But in practice, you sort of get this kind of thing. As data increases, error goes down, and then it sort of stays low. Doesn't go to 0, but it's not high. That's a typical thing in practice. Okay, so, but let's go back to this model. Since we know we have a hierarchical model, how about just invert it as an estimation method? If you invert it, the first thing you're going to do is take the alignments, assuming you know the alignments, which of course is a big assumption, but take the alignments, estimate gene trees, take the gene trees and estimate the species tree. Makes sense, right? Each of them is under a different model, so you can do this. So you can do model-based inference of gene trees, model-based inference of the species tree. To do the model-based inference of the species tree, you need to have some idea about what is the model for how gene trees are different from the species tree. But this is well established for incomplete lineage sorting. So Allman and Degnan and Rhodes did this beautiful theorem that said under the multi-species coalescent, if you only have 4 species, the most frequently observed gene tree is the species tree with probability going to 1. So if you only have 4 species, you don't have to worry about it. Just look at all the gene trees you have on those 4 species. Whichever one appears the most often, these are unrooted gene trees. Whichever one appears most often, that's it. So that gives you an estimation method for 4 species. But to do more than 4 species, you're just combining estimates on quartet trees. They also proved, perhaps in another paper, that it's not true for 5 species because there's something called the anomaly zone, but it's okay for 4. Okay, so Astral is now a method that takes advantage of that quartet theorem. There are other methods that also take advantage of the quartet theorem, but Astral is the one that has dominated the usage now. And the idea is it's taking the gene trees, which are the script T, all your gene trees, and it's finding the species tree that maximizes the quartet support from the gene trees. Really just looking at induced quartet trees. This is an NP-hard optimization problem. Astral is not solving the optimization problem, but what it is doing is solving a constrained version of the problem. So what it does is it says, I'm going to find the optimal solution to this NP-hard optimization problem within a constrained search space. It defines the constrained search space from the input gene trees. It does that in a way that maintains statistical consistency and becomes polynomial time. And the result is that it's low degree polynomial time. So the set of allowed bipartitions is X. The runtime is quadratic at worst in X times the input size. So low degree polynomial time, it's used all over the place. This is just a sample from many years ago. This is the important comparison. This is an analysis on simulated data that Erin Molloy, my former student, did. Only 26 species, but 1,000 genes. And what you see is different methods. Astral is in blue, concatenation using maximum likelihood with Raxamel is in purple. This is the rightmost one. This is Astral. This is error. So low amounts of ILS, high amounts of ILS, very high amounts of ILS. If you compare the methods across the different conditions, you'll also see mean GTEE. GTEE is gene tree estimation error. So you have true gene trees, you have no problem. If you have estimated gene trees, you can have more error. But I just want you to see some examples. Under low ILS, there's really no reason to not do concatenation. Concatenation, purple at the extreme right, is always the best. If you don't have much ILS, you don't need to do anything special. With high ILS, there's now an advantage to using one of these methods, Astral, Astrid, and MPS, 3 different methods that combine gene trees. But when you get to high gene tree estimation error over here, even with high ILS, you might prefer concatenation. And then when you have very high ILS, there's actually more reason to do these Astral and other methods than concatenation until you get to high gene tree estimation error. So this is really interesting. Raxamel is better than these other methods, even though there's no theorem of statistical consistency, even under very high ILS where the anomaly zone is going on. So there is this issue about if you're going to choose a method, it depends upon the data. There's no theorem that can guide you, okay? It depends upon the data. Okay, summary of the lessons from ILS-based species tree estimation. For every 4 species, the most probable unrooted gene tree is the species tree. Astral takes advantage of that in a computationally efficient way by constraining the search space. But concatenation can be a competitive method. It can be more accurate. Those are the lessons about ILS. But what about gene duplication and loss? So we got rid of a lot of data with the 1,000 plant transcriptome project. Gene family trees are what you get when you have gene duplication and loss. If you don't limit yourself to orthologous genes, you get these genes that are multi-copy. So you get a tree something like this. The true species tree has just 3 species, A, B, and C, but there's a duplication up here. So here's the gene family tree. You can see that there's 2 copies of A, 2 copies of B, and 2 copies of C. If that's your input, how do you work with it? So options, you throw out your multi-copy genes. That's what we did with the 1KP project. You figure out orthology. That is not easy. It is not solved. Or you figure out a way to use your gene family trees without knowing orthology. And this is what I'm going to tell you about this. But nothing was proven statistically consistent until 2019. This is pretty recent. So what happened in 2019 is that Sebastian Roch and his student Ben Legrit proved in a paper that we wrote together that the most probable quartet tree under GDL is still the species tree. Same theorem as for ILS. And therefore, a version of Astral that had been designed to deal with multiple alleles actually could be used in this context. It's called Astral Multi, and it would be statistically consistent. That was the first proof. But then Erin Malloy did an analysis comparing Astral Multi. Here it just says Astral, but it's Astral Multi. This is error, this is runtime. Astral Multi to other methods. The only method that it's better than reliably is dupe tree, which is like a parsimony style thing, minimizing duplications. These other methods are not, in fact, provably statistically consistent, but they're more accurate. MolRF and fast MolRFS, more accurate than Astral Multi under all of these settings. So here we have a statistically consistent method that's not as good as something that we don't know if it's statistically consistent. That's a general trend in phylogeny, okay? Statistical consistency says nothing about performance on finite data. It gives you confidence about if you have enough data, nothing about what happens with finite data. Okay, so what was nice is that that result about Astral Multi being statistically consistent got Siavash Mirarab, who is the main developer of Astral, to make a version of Astral that was specifically designed for dealing with GDL. So the idea here is that it takes every multi-copy gene family tree and it roots the gene family tree and it tries to figure out where the duplications occur. And after it's done that, it then sort of modifies the quartet optimization approach to come up with a method that's still polynomial time. Okay, so there's a theorem that says if it correctly roots and tags each gene family tree, it's consistent. But effectively, that's the same as knowing orthology. But that's the only theoretical guarantee. What we saw is that Astral Multi is actually worse than Astral Pro, okay? Astral Pro is more accurate than Astral Multi. It's right here. They're very close, but Astral Pro is better. Fast MolRFS is worse. But runtime-wise, Astral Pro is pretty good, not quite as good as Astral Multi. So here you have it, a method that's actually practically useful. It might be statistically consistent even with errors, but we don't have a proof. Okay, two key ideas for, remember this is from what I showed you before, the two key ideas from ILS. The idea that every 4 species, the most probable gene tree is identical to the tree on the true species tree, and that Astral can find solutions using this constrained optimization. It's the same thing for ILS plus GDL. Same thing, the most probable unrooted gene tree is topologically identical to the true species tree, even when you have ILS and GDL together. And the same algorithmic approach can work, but for statistical consistency. And Astral Pro, although it's not guaranteed statistically consistent, it has this additional step, rooting and tagging gene trees, okay? So what about HGT? There's actually other theorems by Sebastian Roch and Sigi Sneer that show that the most probable quartet tree is equal to the species tree here. So if you think about lots of HGT, but there's still an underlying species tree, can you get the underlying species tree? They proved that that was possible with quartet trees, with random HGT, but bounded amounts. Sigi Sneer and Sebastian Roch. So we actually did a simulation and explored this, and we showed that quartet-based methods like Astral were more accurate than concatenation. So we got this thing over and over again. Quartet methods can give you good results. It's not true under every model of gene tree discordance, but under multiple models. Okay, so idea, you take gene trees, you get species trees. You do it with quartet trees. Quartet-based tree estimation can give you very good results. Not mentioned here, it's much faster than concatenation. You can replace the 250 CPU years that we did for the Avian tree, 250 CPU years for a concatenation analysis with something that just takes perhaps minutes. There's really a big difference in runtime. Okay, part 2, I'm going to zip through part 2 and part 3. All of this, Avian was 48 species, the 1KP was like 100 species. What happens if you want to get a tree on 1 million species? We have heard that there are many, many species on Earth. How do we get trees that are really on hundreds of thousands of species, okay? Again, we're getting unrooted trees. There are these models of sequence evolution. You can see there is no indels here, it's just substitutions. There are sequence evolution models such as Jukes Cantor, the generalized tree, generalized time reversible model, GTR. There's many models of substitutions for sequences evolving down trees. They all have more or less the same flavor, IID across the sites. You have something going on here. You just described it with some parameters. Maximum likelihood is very, very popular and provably statistically consistent under the nice models. Nice models are ones that are IID. Okay. I'm going to talk about results with maximum likelihood because that's what people do with their data sets that are big. The ones who have the time can do Bayesian. In theory, consistent, low sample complexity, and empirically, you get really good results even with heuristics. You're not provably solving your problems, you're running heuristics. RAxML is the standard one. But RAxML is not designed for very large trees. So we came up with a technique that's divide and conquer that allows you to take a large data set, divide it into disjoint sets, get trees on the disjoint sets, and then merge the trees. It's not a supertree method because these are disjoint. And by putting RAxML on the subsets, you can actually take the advantage of RAxML's accuracy, but limitation to small trees. So here are results that we published on data sets that range from 1,000 sequences to 50,000 sequences. This is tree error. So RAxML is here in red. This is IQ tree. This is fast tree, IQ tree, RAxML, and here's our divide and conquer technique. And what you can see is fast tree is terrible here. RAxML is the best. This is a little bit worse. They're all about the same. Fast tree is bad. These are the same. Fast tree is actually not bad. RAxML is worse. Our divide and conquer is better. And here we can only run fast tree and our divide and conquer technique. So focusing just on the data sets where, the really large data sets with 10,000 and 50,000, you can see that we can get trees that RAxML cannot even analyze. So this is a very recent work that we're still trying to explore. So if you have very large trees, you want to get maximum likelihood trees, this is something you can try. But you can use this with any kind of tree estimator, not just RAxML. You can use it with species trees. It actually improves accuracy and reduces runtime. So it's a general purpose divide and conquer technique for large trees. Large in this case is the number of leaves in the tree. Okay, my last part, which is where I want to leave you with a conundrum. Multiple sequence alignment. You can't do anything here without a multiple sequence alignment. How do you get a good multiple sequence alignment? So multiple sequence alignment is actually one of these really big grand challenges. It has applications in many areas. There's hard optimization problems, but the current many methods that are very good just don't scale to large data sets. By the way, Kato spoke 2 days ago, and he's the developer of Math, which is really like the best off-the-shelf software for multiple sequence alignment, but it doesn't really scale well to large data sets. Alignment is actually recovering history that's evolutionary. If you have a sequence that evolves into another sequence with insertions and deletions and substitutions, you can line them up. So this stuff got deleted. So this stuff is above dashes. This T changes into a C. So the T is above a C. This T was inserted, so it's below a dash. Pairwise alignment is a reflection of evolutionary history. Multiple sequence alignment is the same thing, just with transitivity. So you have this thing, the true pairwise alignment is recovering the evolutionary history. What makes alignments difficult? Large numbers of sequences, high rates of substitutions, high rates of insertions and deletions, very long sequences, and sequence length heterogeneity that can be the result of evolution or of technology, like if you're including reads into your data set. So we did a study, this is back from 2009, comparing different alignment methods with RAxML. When you do a simulation, you know the truth. So you can compare the true alignment to the estimated alignment, the true tree to the estimated tree. this is, all these data sets have 1,000 sequences, 15 different models. The top is tree error, the bottom is alignment error. As you go from left to right, it's more or less sorted so you can see errors increasing. What's really happening as you go from left to right is that to some extent, the amount of evolution is going up. Substitutions are going up, indels are going up, but there's different lengths for the indels. So what happens as you go from left to right is XML in the true alignment is in black. So there's tree error is low here, then it goes up a little bit. And it doesn't matter how you align the data sets for the first 6 models because you get the same tree error. But there is difference in the alignment error. And then as you go to higher rates of evolution, the tree error goes up just because of the rate of evolution. But if you have to estimate the alignment, error goes up for the alignment as well. And the one thing to notice is that Clustal, which is like the most commonly used alignment method, has the worst accuracy on large evolutionary distances. Okay, so what's going on here is that some data sets with 1,000 sequences are easy. Some are hard. What matters is the evolutionary diameter. So this suggests a divide and conquer strategy. Take your data set, divide into subsets that are lower diameter, get alignments on the subsets, merge the alignments. When you've done that, you can get a new tree, and then you could iterate. And if you iterate, each iteration might improve the alignment, therefore improve the tree. So we studied that in 2009 with this method called SATE. Here's the result of a 24-hour analysis with SATE. Better alignments, better trees. That led to other developments of the same type of thing, iteration with divide and conquer. And the last methods of this sort can go to 1 million sequences with very high accuracy. So basically, if you have a large data set, you don't have to give up multiple sequence alignment. You just have to use something that's not off the shelf. One could say PASTA is off the shelf, but PASTA, it's been around for a long time. Magus is now better than PASTA. And it's using MAFT on subsets. MAFT Lindsay is great, but it doesn't go to large data sets. Okay, statistical alignment. Valified by Ben Redlings and Mark Suchard was a co-estimation method for statistical alignment. It's the main method for this problem, but it's limited to small data sets. And small meaning they suggest nothing more than 100. Here's a simulation on proteins, only to at most 26 sequences. Each Balify analysis took 2 months of runtime. The others take a few minutes. What you're looking at here is Mahler score and SP score. This is essentially precision. This is recall. The closer you are to the top corner, the better. This is low rate of evolution of indels. This is, and these are substitutions and indels. This is the hardest condition. This is the easiest condition. The red dot is the one that's closest to the best. On simulated data, it's the best. Okay, Balify is the best, except different story on biological data. These are different protein structural benchmarks, same data set sizes. And what you can see is the red dot is no longer at the top. Now, this is the recall, this is precision. It's just not great. So this is where I'm leaving you with a conundrum. I don't know what's going on. I don't know why we're seeing this. Every time I ask someone what they think is going on, they give me a different answer. But 3 possible explanations. Model misspecification, i.e. the Balify model is just not biologically realistic for proteins. Another one is that structural alignments, which is what the benchmarks are actually very different from evolution. I don't think they're so different that it would explain everything. And then the other one is that the structural benchmark alignments are simply not correct, even as structural alignments. That's possible. But it depends on who you ask, they have a different answer. So this is my question, right? And the reason I think this is important is that if this is the problem, model misspecification, then what aspect of the model is wrong? And that will impact what we do in everything. And if it's just that the benchmarks are wrong, okay, fine. That's not so worrisome to me. Okay, and with this, I want to thank you for your attention.
One question.
Thank
you for that talk, Tandy. I'd like to ask you about your last slide and the 3 points you raised. I sort of have a interest in hoping that it's number 1, but I'm actually going to ask about your comment about number 2. You said that you thought you could see structural alignments and evolutionary alignments being different, but you didn't think that would explain everything. I wondered if you could elaborate on that a little bit.
Okay, so the point is we know that structural alignments can be different from evolution because you can have parallel evolution, you can have convergent evolution to have certain structures. Does it happen so frequently that you'd get this kind of really significant difference? That's the question. Not does it happen, but does it happen so much that it would explain that? Right? So of course, there's some people who would say it does happen, but it doesn't happen enough to explain it. Other people would say this is the major explanation. So actually, didn't we have this conversation? Didn't I already ask you what's your answer?
Well, I mean, I think that, you know, when you get recurrent insertions and deletions in loop regions in proteins, then if you do a structural alignment, it'll look like they're in the, you know, these residues are aligned.
So you think that that would be explaining all of these, every single benchmark here?
I mean, I think it'll give you distorted estimates of, you know, indel rates and over alignment in loop regions and proteins. I don't know if it's enough to explain all your.
Okay, well, I think the thing to realize here is what Balify is doing is it's under aligning. So the structural benchmarks look over aligned relative,
yes, right.
But the over alignment could be a problem about the structural alignments. It could be more this one. Because the structural alignments are done by humans, not just by structures, right? And this is where Nick Goldman says they always like to see things lining up.
Exactly.
So that's not quite the same thing as structural alignments and evolutionary alignments being different. It's more that humans want things to be lining up.
I see.
Okay, anyway, as you can see, there is this conundrum. I don't know the answer. And I'd love to have people tell me over the coffee break what they think is going on. That's
Thanks.
it, right?
Okay,
Thank
thank you
you.
very much.
The next speaker.